Welcome, curious minds, to the deep dive. Today, we're plunging headfirst into a microscopic world that honestly might completely reshape how you think about some really infamous plant diseases in history. Here's a thought for you. What if we told you that the uh, devastating Irish potato famine wasn't actually caused by a true fungus at all?
Yeah, that's pretty surprising.
Imagine these organisms that look, act, and even spread like fungi, but genetically speaking, they're actually closer to, well, algae.
It's genuinely one of the most intriguing reclassifications in biology. It really is.
Yeah.
For such a long time, these organisms, the Oomycota, were grouped right in there with what we typically think of as fungi.
Right.
But as our scientific tools got better, especially with modern genetic analysis, their real family tree became super clear.
Yeah.
And it wasn't with the fungi.
And that's exactly our mission today on the deep dive. We're taking your sources, focusing especially on insights from a chapter in Introductory Mycology to sort of untangle this curious and, frankly, profoundly impactful world of Oomycota. You might hear them called water molds sometimes.
That's a common name, yeah.
We're going to pull back the curtain on their really unique structure, their surprisingly varied ways of reproducing, their distinct inner workings, their physiology and genetics, and how they fit into the bigger picture, the tree of life.
And crucially, their huge ecological and even medical significance.
Exactly. Our goal is for you to really grasp every key concept, understand why it all matters, and feel totally informed without getting, bogged down. So let's unpack this biological mystery. Okay, so our journey kicks off with this big reveal. For centuries, Oomycota were just mistakenly lumped in with true fungi. You see, they look incredibly similar, especially in how they absorb nutrients. From the outside, very fungus-like.
Totally. It was a classic case of convergent evolution leading to this kind of mistaken identity. Early mycologists looked at their stringy filamentous growth, how they fed, and, well, it just seemed logical to put them together.
Made sense at the time.
It did. But even way back in the 1800s, some really pioneering scientists, like Pringsheim and later others like Kreisel and Schaffer, they started noticing little differences, things that didn't quite fit the fungal mold, so to speak.
Ah, early clues.
Early clues, yeah.
Yeah.
Fast forward to today, and the genetic evidence, it's just irrefutable. Oomycota share basically no close evolutionary relationship with the true fungi. None at all.
So if they're not fungi, who are they related to? This is where it gets really cool, I think. It changes how we see the biological family tree.
It really does.
You might be surprised to learn their actual closest relatives are the heterocont algae. Those often brown or golden brown algae, the ones with chlorophyll A and C?
Exactly, which means Oomycota are now firmly placed within the kingdom Streminopola. It's this wonderfully diverse group that includes everything from, like, giant kelp all the way down to these microscopic plant destroyers.
Wow. Okay, so what really sets them apart then, if you look closer?
Well, it's remarkable how clearly different they are once you get past the superficial stuff. Take their unique asexual spores, for instance. They're called zoospores.
Zoospores, right.
Unlike true fungi, these are biflagellate. That means they have two distinct whip-like tails or flagella.
Okay, two tails. How are they different?
Picture one longer, tinsel flagellum. It's covered in these tiny fine hairs, and it sort of pulls them forward. Then there's a shorter, smooth whiplash flagellum that trails behind, pushing them. This whole setup, it's totally unique to them. A dead giveaway.
Like a little microscopic motor with a rudder?
Kinda, yeah. And it gets even more fundamental. Their main body, the phallus, it's typically diploid.
Diploid meaning two sets of chromosomes, right? Like us?
Exactly. Two full sets in almost every cell of their main body. This is a huge contrast to most true fungi, whose main body is usually haploid, just one set of chromosomes, or sometimes dikaryotic, which is different again.
So for Oomycota, the process of meiosis, where the chromosome number gets halved, that only happens in their special reproductive bits.
Precisely. Only in the gametangia, their reproductive organs.
Okay, what about the cell walls? That's usually a big fungal thing, the chitin.
Right. True fungi are famous for chitin walls, same stuff as in insect shells. But Oomycota, nope. Their walls are primarily made of beta-glucans and some cellulose.
Cellulose, like plants.
Like plants, yeah. Now, interestingly, a tiny, tiny amount of chitin has been found in a few species, like Achlea and Saprolania, but it's definitely not their main building block. Plus, they have an amino acid called hydroxyproline in their walls. Another difference.
And even inside the cell, different machinery.
You bet. Their mitochondria, the powerhouses, have tubular cristae. Those internal folds look like little tubes. True fungi. They have plate-like cristae, more like stacked pancakes.
Different plumbing.
Different plumbing. And their Golgi bodies, the protein packers and shippers. They're quite complex, with multiple flattened sacs, or cisternae, much more elaborate than the simpler Golgi you find in true fungi.
And lastly, how they reproduce sexually.
Also distinct. They typically use something called oogamous reproduction. It involves direct contact between their reproductive structures, the gametangia. This leads to a really tough, thick-walled sexual spore called an oospore.
So yeah, these aren't just minor tweaks, they're fundamental biological differences.
Foundational differences, exactly. Shows a completely separate evolutionary path.
So what do these not-so-fungi actually look like? You mentioned filamentous.
Yeah, their body plans can vary quite a bit. Some are single-celled, but many form these complex filamentous structures. Often they grow as profusely branched, coenocytic hyphae.
Coenocytic. Remind us what that means.
Sure. Imagine their main body threads, the hyphae, as long, continuous tubes. There are no internal walls or septa dividing them to separate cells. It's like one big, interconnected cytoplasmic highway filled with lots and lots of nuclei, all flowing together.
So like an open-plan office for nuclei.
Huh. Yeah, something like that. Any walls you do see are usually just at the base of the reproductive structures or maybe in older parts that are getting walled off.
And how do they grow and eat? You said absorptive nutrition.
Right. Like true fungi, their hyphae grow at the very tips. There's a whole bunch of tiny sacs, apical vesicles involved in building the new cell wall at the growing point. But
Yeah.
the parasitic ones, they've got some special tools.
Ah, the nasty bits.
The nasty bits for the plant, yes. They're called haustoria. Picture these tiny specialized branches that could be peg-like or spherical or even lobed that the seat pushes into the host plant cell.
Okay. Do they just punch through?
They penetrate the host cell wall, but here's the clever part. They don't usually rupture the host's plasma membrane, the delicate layer just inside the wall. Instead, they invaginate it. They push it inwards, creating this little pouch.
Ah, so they're inside the cell wall but wrapped in the host's own membrane.
Exactly. It allows them to absorb nutrients directly from the host cell cytoplasm without necessarily killing it immediately. It's a very sophisticated way to parasitize.
Wow. Okay, and you mentioned unique internal features too, fingerprint vacuoles.
Yes, my personal favorite as well. They have these things called fingerprint vacuoles or dense body vacuoles. And under the microscope, they genuinely have these intricate internal patterns that look like, well, fingerprints.
That's wild. Any idea what they're for?
The best guess is they might be storing polysaccharides, probably their storage compound, mycolaminarin, inside them.
And lots of nuclei. Push this out.
Yeah, as the hypha grows, a large central vacuole often forms behind the tip, and it can push the numerous nuclei towards the edges of the hypha. And one more internal detail. Their cell division, both mitosis and meiosis, is intranuclear and centric.
Meaning
Meaning the nuclear envelope, the membrane around the nucleus, stays completely intact during division. All the action happens inside. And centrioles, which help organize division, are present. This is different from many other eukaryotes, including fungi, where the nuclear envelope often breaks down. Subtle, but significant.
Okay, let's get into the reproduction, because this seems like a key part of their success, right? This sort of double life, asexual and sexual.
Absolutely. They're masters of propagation. Asexually, it's all about those motospores we mentioned, the zoospores, and they actually make two different kinds.
Two types.
Yeah. First, you have primary zoospores. These are often pear-shaped, and their flagella are right at the pointy anterior end. They tend to be, let's say, less impressive swimmers, maybe a bit clumsy, considered a more primitive type.
Okay, the learner permit swimmers.
Yeah. Then you have the secondary zoospores. These are much more common and way more effective. They're typically kidney-shaped or bean-shaped. And their flagella, they're inserted on the side, laterally, in a groove.
And they point outwards?
Usually away from each other, yeah, at about a 130-degree angle. That longer, hairy tinsel flagellum leads pulling them forward, while the shorter, smooth whiplash one trails, pushing. They're powerful swimmers.
Got it. And these develop inside?
Inside sac-like structures called sporangia. When they're mature, the sporangia release them, usually into water. They swim around, but typically only for short distances, actively looking for a host or something suitable to grow on.
And when they find a spot?
They insist, quickly. They ditch the flagella, pull themselves into a round shape, and form a thin protective wall. After a short rest, they germinate. A little tube, the germ tube, grows out, and that develops into new hyphae. Boom, new infection or colony.
It sounds efficient, but how do they find the right spot, especially the plant parasites?
That's where chemotaxis comes in. They're chemically attracted. They can sense specific chemical signals, exudates, released by plant roots or other hosts. It guides them.
Like little heat-seeking missiles, but for chemicals.
Exactly. A great example is Pythium disodicum. Its zoospores are known to swim towards cotton roots and specifically accumulate and insist right on the root cap cells. Nowhere else. It's incredibly precise targeting.
That's amazing. Microscopic GPS.
Pretty much. And then sometimes you find truly bizarre adaptations like Haptoglossa mirabilis. It's an umiseed that parasitizes tiny aquatic animals called rotifers. Its encysted spore develops into this specialized structure called a gun cell. And it literally, I mean literally, shoots a harpoon-shaped projectile into the rotifer, and then it injects its entire cellular contents through that projectile.
They're kidding.
Yeah.
A microscopic harpoon gun.
No kidding. It's an incredible mechanism, a biological cannon for infection.
Wow. Nature's wild. But do they all make these swimming spores?
Good question. No, not all of them. Some groups have lost the ability. They're called aplonetic. And many plant parasites, especially the more advanced ones, have evolved a shortcut.
A shortcut?
Yeah, they produce structures that look like sporangia but function like conidia, like fungal spores. These conidium-like sporangia don't release zoospores. Instead, they just germinate directly by putting out a germ tube. Often, whether they release zoospores or germinate directly depends on the environment, like temperature or whether there's free water around.
Clever adaptation. Okay, what about the sexual side of things? The oospore.
Right, sexual reproduction. It's almost always heterogametangic,
Right.
meaning you have distinct male and female structures interacting.
Yeah.
The male parts are called antheridia, and the larger, usually spherical female parts are the euthegonia.
And this is where meiosis happens.
Yes, inside these structures. Meiosis occurs, producing haploid nuclei. Then typically a delicate fertilization tube grows from the antheridium over to the oosogonium. It penetrates the oosogonial wall and delivers a male nucleus to fuse with a female nucleus inside an egg cell, or oosphere.
And that fusion creates the diploid oospore.
Exactly. Karyogamy, the fusion of nuclei, happens, restoring the diploid state. And that fertilized oosphere matures into the thick-walled, super-resistant oospore. It's built to last, to survive tough times like winter or dry periods.
Now, wasn't there something about hormones controlling this in some species?
Ah, yes. Really fascinating stuff, discovered in Agaricales species. Groundbreaking work showed they use steroid hormones, like actual steroids. One called antheridiol is released by female hyphae and triggers the formation of male branches, the antheridia. Then the antheridia produce another hormone, euganiol, which induces the formation of the female eugaridia.
Wow, like a chemical conversation coordinating everything.
Precisely. It's this sequential, hormonally regulated development. Finding steroid hormones acting like this outside the animal kingdom was, and still is, a huge deal. Shows a surprising level of chemical sophistication.
Okay, moving beyond structure and reproduction, let's talk biochemistry, because you said this is another area where they really differ from true fungi.
Absolutely, some really fundamental metabolic differences. Take how they make the essential amino acid lysine.
Okay, lysine. Important building block.
Very. Mycota synthesize lysine using something called the diaminopomalylic acid pathway, or DAP pathway. Now, the key thing here is that's the same pathway plants use.
And fungi.
True fungi use a completely different route. The alpha-aminodipic acid pathway, AAA pathway. It's like they follow entirely different biochemical roadmaps to make the same crucial molecule.
That's a really deep difference.
It is. And here's another one, crucial for disease control. Sterols.
Sterols. Like cholesterol?
Sort of related, yeah. Sterols are vital components of cell membranes. Now, true fungi typically make ergosterol. Many common antifungal drugs work by targeting ergosterol synthesis.
Right, okay. So what about Mycota?
They generally don't make ergosterol. Some can make their own sterols, others need to get them from their environment, but their characteristic sterol, if they make one, is fucosterol.
Fucosterol, not ergosterol.
Exactly.
Which makes
Most ergosterol targeting fungicides won't work on them.
Bingo. That's a major reason why controlling umiseed diseases often requires different chemical approaches than controlling true fungal diseases. Understanding their unique biochemistry is critical for effective management.
makes perfect sense. What else is biochemically unique?
Their main storage carbohydrate. True fungi typically store energy as glycogen. Mycota primarily use mycolaminaran.
Mycolaminaran. Sounds different.
It's a water-soluble beta-1-3 glucokin. And again, what's interesting is that it's similar to storage compounds found in, guess what, certain algae. Reinforces that connection.
Another link back to algae.
Yep. And finally, they also lack certain sugar alcohols, acyclic polyols. They're pretty common in true fungi. So layer upon layer of biochemical differences really sets them apart.
Okay, so we've established they're unique structurally, reproductively, biochemically. Now let's talk impact. Why should we care so much about these not quite fungi?
Well, their impact is huge, both ecologically and economically. Let's start with ecology. Many aquatic Mycota, those classic water molds, are incredibly important
Dream
saprobes.
composers, right. Breaking down dead stuff.
Exactly. In freshwater and saltwater ecosystems, they play a major role in breaking down dead plants and animals, recycling nutrients. Essential work. But they're not just recyclers.
They're parasites, too.
Oh yes, they parasitize a whole range of aquatic critters. Tiny rotifers, nematodes, mosquito larvae.
Ah, mosquito larvae. Potential there.
Potential for biocontrol, yes. Also crayfish, fish. They can cause significant diseases in aquaculture.
But the really big impact, economically speaking, is on plants, isn't it?
Absolutely devastating. They are some of the most important plant pathogens known. The most infamous, the poster child, has to be Phytophthora infestans.
Late blight of potato, the Irish potato famine.
The very one. Caused unimaginable suffering in Ireland in the mid-1800s. Mass starvation, mass immigration. It literally changed the course of history, all because of this one microscopic seed.
How does it work? How does it spread so fast?
It survives between seasons in infected potato tubers left in the ground or used as seed.
Mm-hmm.
Then, under cool, wet conditions, which Ireland often has, it takes off. Specialized branches called sporangiophores grow out through the stomata, the breathing pores on potato leaves.
And release spores.
They release those sporangio we talked about. They can be splashed by rain or blown by wind to new plants. If there's moisture, they can release zoospores or germinate directly, starting new infections very, very quickly. An entire field can be wiped out in days.
Just devastating. And it's still a problem.
Still a major global problem, and made more complex by the spread of different mating types, A1 and A2, which allows for sexual reproduction and generates new genetic diversity in the pathogen.
Wow. Okay, another big one you mentioned in the outline was downy mildew on grapes.
Ah, yes. Plasmopara viticola. Another great story, in a way. It causes grape downy mildew. It was accidentally introduced from North America to France in the late 19th century.
And nearly wiped out the French wine industry, I gather.
Pretty much. It was a catastrophe. But this disaster led directly to the discovery of the first really effective fungicide, Bordeaux mixture.
How did that happen? Accidentally?
Totally accidentally. A professor named Millardet noticed that grapevines along the roadside, which had been sprayed with this bluish mixture of copper sulfate and lime to stop people from stealing the grapes.
It does look poisonous.
Exactly. He noticed those sprayed vines were mysteriously free of the downy mildew that was ravaging nearby vineyards. He put two and two together, experimented, and voila, Bordeaux mixture was born. A landmark moment in plant protection.
Amazing. Necessity, or rather deterring theft, is the mother of invention.
Something like that. And we can't forget Pythium species. They cause damping off.
Damping off. Sounds gentle, but I bet it isn't.
Not at all gentle for seedlings. It's when young seedlings suddenly collapse and die, either just before they break the soil surface or shortly after. Pythium deberianum is a classic culprit. It's a huge problem in greenhouses, nurseries, and fields worldwide. Very costly.
And you mentioned one, Phytophthora cinnamomi, that attacks loads of different plants.
Yeah, that one's a real beast. P. cinnamomi is known to cause disease in something like 1,000 different plant species around the globe. Avocado root rot is a big one, but it affects forests, nurseries. It's incredibly widespread and destructive.
It's not just plants, though, right? You mentioned medical relevance?
Correct. Less common, but still significant. Pythium insidiosum causes a disease called pythiosis. It's a serious condition, often forming granulomas, these sort of nodular lesions.
In humans?
Primarily in mammals like horses, dogs, and cattle, especially in tropical and subtropical areas. But yes, occasionally it can infect humans too. It's often difficult to treat because, again, it's not a true fungus, so standard antifungals might not work well.
A sobering reminder they cross kingdoms. But there was a positive biocontrol angle, too.
Right, with Leginium giganteum. It specifically parasitizes mosquito larvae. So there's active research into using it as a biological control agent, maybe reducing the need for chemical pesticides to control mosquitoes.
Interesting. And something about shrimp and bacteria.
Oh yeah, that was a cool interaction. Laginidium calinex can parasitize marine crustacean eggs and larvae, like shrimp. But researchers found that a specific bacterium living on the surface of shrimp eggs produces a chemical, isatin, that actually inhibits the oomycete.
Wow, a protective bacterial shield.
Basically, yeah. Shows the incredible complexity of microbial interactions out there.
Okay, let's talk evolution. You mentioned these two main lineages, these sort of galaxies.
Yes, that was Frank K. Sparrow's idea, a real giant in the field. He talked about the Saproleniomycetidae, the Saprolenian galaxy. These are mostly your classic aquatic water molds, generally considered the more primitive or ancestral line,
And the other galaxy?
the Peronospora or the Peronosporation galaxy. This group contains mostly terrestrial species, and especially those highly specialized plant parasites, like Phytophthora and the downy mildews. They're seen as more derived, more highly evolved in terms of their parasitic adaptations.
And as they evolved along these paths, what were the major trends? What changed?
We see some pretty clear shifts. There's a major trend moving away from being purely saprobic, just eating dead stuff, towards facultative parasitism, being able to switch between dead stuff and living hosts, and then finally, in the most specialized groups, becoming obligate parasites. They have to have a living host.
Makes sense. Specialization.
Exactly. Linked to that, some groups lost the ability to make their own sterols, becoming dependent on their host. We generally see hyphae getting thinner, more delicate, perhaps better for navigating plant tissues. And the development of those specialized feeding structures, the haustoria, becomes much more common and elaborate in the plant parasites.
What about reproduction? Any trends there?
Yes. There's a trend towards losing the primary zoospore stage, and in the more advanced terrestrial forms, like some downy mildews, there's a tendency for the sporangia to just germinate directly with the germ tube, skipping the swimming spore stage altogether. Perhaps an adaptation to drier, wind-dispersed conditions.
And specialized structures for dispersal.
Right. The development of those distinct sporangiophores, structures designed to hold the sporangia up for dispersal, often by wind, becomes prominent in groups like the Peronosporaceae. Yeah. And in sexual reproduction, we sometimes see a reduction in the number of eggs, oospheres, per oegonium, or the number of antheridia involved, and a shift towards heterothallism.
Heterothallism meaning they need two different mating types, like male and female strains, to reproduce sexually.
Precisely. Requires outcrossing, which can boost genetic diversity. So it's a whole suite of adaptations driven by the shift towards terrestrial life and specialized parasitism.
Okay, maybe we can quickly touch on some specific examples from the different orders just to put some names to these trends.
Sure. The Saproleniales, these are your classic water molds. Many are easy to isolate, grow in the lab. Think Saprolenia itself, often seen on dead fish or insects in ponds. Aphanomyces astacei is in this group, causes the devastating crayfish plague. Some, like Saprolenia, are dimorphic with both primary and secondary zoospores. Others, like Aclea and Aphanomyces, are monomorphic, sipping the primary type.
And Aclea also had those hormones, and Gemmae.
Right. Aclea reproduces asexually, not just with zoospores, but also with gemmae, basically just modified bits of hyphae that break off and can start a whole new colony. Very effective.
What about Leginidiales?
Mostly aquatic parasites. We mentioned Leginidium calinex on crab eggs and L. giganteum on mosquito larvae, often parasites of algae or other fungi too.
And Leptometales. Anything special there?
They often have constricted hyphae with little plugs called cellulin granules. And interestingly, for oomycetes, they do seem to have a noticeable amount of chitin in their walls, along with cellulose. A bit unusual.
Rapidiales. Sound obscure.
They are a bit unusual. They tend to grow in polluted, stagnant water with low oxygen. Some, like Aquilindorella fermentans, are even facultative anaerobes. They can grow without oxygen. Quite different.
Okay, then the big one. Peronospiralis. The major plant pathogens are here.
Absolutely. This order is split into a few key families. The Piceaceae includes Pythium and Phytophthora. Their sporangia might release zoospores directly, or sometimes into a little vesicle first, or they might just germinate directly. Remember Phytophthora infestans and its weird amphigynous development where the ogononium grows through the antheridium?
Yeah.
That's characteristic of many Phytophthora.
Right, and the downy mildews.
They're in the Peronosporaceae. Think Plasmopara, grape downy mildew, Peronospora, Bromea, Lett's downy mildew. Key features are those distinct, often branched sporangiophores that have determinate growth. All the sporangia mature around the same time and get released, usually by wind. And as we said, some advanced ones like Peronospora just have the sporangia germinate directly. Obligate parasites, these guys.
And finally, the white rusts.
The Albuginacea. The main genus is Albugo. They cause white rust diseases. You recognize them by their characteristic chains of sporangia formed under the host's epidermis on short, club-shaped sporangiophores. Eventually, the epidermis ruptures, releasing a white, powdery mass of sporangia. Albugo candida on cabbage and relatives is a very common example. Also obligate parasites.
Wow. Okay, so we've really covered a lot of ground. We've journeyed through the really intricate biology of Omicota, their unique cells, their diverse ways of reproducing those distinct biochemical pathways.
Yeah, and we've seen their vital roles as decomposers in water, but also their absolutely formidable impact as plant pathogens literally shaping human history in some cases.
And even popping up with some surprising medical relevance, too.
Indeed. It's just a powerful reminder, I think, that biological labels can sometimes be misleading. And really, some of the most profound discoveries happen when we challenge those old classifications and look deeper. The more we understand these fungi that aren't fungi, the better we can manage their impact, good and bad.
So next time you happen to see maybe a fuzzy patch on a decaying leaf in a pond, or you hear about some crop disease causing trouble, maybe remember these imposter fungi. Remember how diving into their world really reveals just how interconnected things are and how impactful even the tiniest organisms can be?
Well said.
It really makes you wonder, doesn't it? What other organisms that seem simple or well understood on the surface might be hiding these incredibly complex stories, just waiting for us to, take a closer look and ask the right questions.
That's the exciting part of biology. Always more questions to ask.
Thank you so much for joining us on this deep dive. We really hope this exploration has given you a newfound appreciation for, well, the hidden complexities all around us in the biological world, and maybe, just maybe, sparked your own curiosity to keep digging deeper.